so we have our strands of DNA going from 3' to 5' on this side, 5 to 3 on that side. They're anti-parallel. Our DNA nucleotides are in white. They're linked together by hydrogen bonds labeled with the pink sticky notes. On the edges here, each individual nucleotide is tied together with phosphodiester bonds, linking from the phosphate group to the third carbon on the sugar. So for the purpose of this video, the RNA primer is going to be 4 nucleotides long. The Okazaki fragment will be 6. So helicase comes in. It's going to start breaking these hydrogen bonds. It's going to be followed by gyrase, which stops supercoiling. Then SSG is going to come in. It's going to coat the DNA strand to prevent new hydrogen bonds from forming. Primase comes in and starts our RNA primer. It's built from the 5' to the 3' direction. So G matches with C, A matches with U, because this is RNA, U replaces T, G matches with C, A matches with T. DNA polymerase 3 comes in. These continue to open up. DNA polymerase 3 comes in. We start our DNA sequence. C matches with G. And G matches with C. So we've got our 6 here. DNA polymerase 1 comes in and replaces these 4 leading RNA nucleotides with DNA. So it starts at the 5' end, G matches with C, A matches with T, G matches with C, and A matches with T. Then DNA ligase comes in and links these 2 DNA fragments together.
So now we can start the RNA replication of the second DNA strand, starting from the 5' end down to the 3' end. And C will connect to G, G connects to C, A connects to U, because in RNA thymine is replaced by uracil. Then G connects with C, and then DNA polymerase 3 comes in, stops the RNA and replaces it with DNA. So T connects to A, and C connects to G, and then DNA polymerase 1 comes in, replaces the 4 RNA nucleotides with DNA nucleotides. So C connects with G, G connects with C, A connects with T, and C connects with G. And then DNA ligase comes in, connects the 2 DNA fragments to create the Okazaki fragment. Yeah.
All right, continuing up, helicase continues to break the bonds. So for the next segment here, A matches with T, C matches with G, T matches with A, T matches with A matches with T, and G matches with C. Now that we have our next 6 open up, we can start the Okazaki fragment on the other side.
So now that helicase has opened up the next 6 hydrogen bonds, we can start our RNA transcription on this side, which C matches with G, T matches with A matches with U, A matches with U, and then DNA polymerase 3 comes in, stops the RNA, and starts DNA. So G matches with C, T matches with A, and then DNA polymerase 1 comes in, replaces the 4 RNA nucleotides with DNA nucleotides. So C will then match with G, T will match with A will match with T, A will match with T, and then DNA ligase comes in, connects the two, and makes our third Okazaki fragment. Go. So now we can take our two Okazaki fragments and connect them with DNA ligase to create one continuous strand.